Okay, welcome everybody to our Sno-King Water Watchers Monitor meeting for February 22nd, 2/2 of 22. What we're going to talk about this evening is we're going to talk about a project we have coming up that is a DNA sampling project, which is a pretty interesting project. We're going to talk about the Global Water Watch database, bacteria counting issues and tips, upcoming monitor training that we have for March and April, which includes water chemistry, bacteria stream biomonitoring, flow monitoring, and habitat assessment as well. And also we're going to have some training along the lines of biomonitoring. We're also going to be talking about BI, and we'll get into that as well. And we're going to talk about assessments for your stream site and the best times to do them, and where and how to get supplies. I think there's one person who might not be muted because I'm hearing some Doritos wrapper crinkling or something. Okay, so let's talk about our DNA sampling project. So first of all, I would, we talk about DNA a lot. I don't know, I shouldn't make the assumption everybody knows what DNA is, but essentially it's an instruction set for building proteins, and it's useful because it's in almost every living cell, and it can be sequenced, and it has a unique sequence that's associated with different types of organisms. And so that's useful for identification purposes. And so in our case, we're going to be using that characteristic of DNA to identify bacteria in streams. And so this is an example of a map where different types of DNA or different DNA profile identified major contaminants as being from particular sources in a watershed in California. And there's a company that we're working with called Veriset that is doing they're the ones that have developed this technology, this phylochip technology, that's going to let us do this very detailed sequencing. So we already know that bacteria can be a problem in water, and that we can use this technique for identifying bacteria sources. And what's different about this than what we normally do is our E. coli testing that we do identifies levels. So we can say, well, it's high or it's low. And if it's high, we know that that might be dangerous for human health, but we can't really say, where does it come from specifically? And so one of our partner organizations, Thornton Creek Alliance, has a citizen science coordinator, Gary Olson, who learned about this particular company offering detailed DNA profiling. And so we're going to go through, we got funding from King County Waterworks to do a project to do some of this for some of our sites. And so what's useful about this particular technique is that it can identify not just at a general level, like is it human or some other type of animal, but it can get down to a very specific level, like if it's from a human source, was it from a leaking side sewer? Was it from a failed septic system? Was it from an encampment, et cetera? So a lot of times jurisdictions have, they're one of the water quality problems that their water bodies are listed for is bacteria contamination. And so theoretically, they're trying to do something to remedy that bacteria contamination. But a lot of times, if they don't know exactly where it's coming from, it's hard to target a particular source. So we're hoping that this could be proven to be useful and target particular sources. Okay, let's talk a little bit about the Global Water Watch database. So it is where we've been entering our data and viewing our historical data. And we've been using this for about the last, 4 or 5 years, their database. We've used Global Water Watch techniques. They have training materials. That's the kind of the source of our program. Part of their program that we've also adopted is their database, where we enter data and we can view that data online. So it's very useful in that regard. it is housed at Auburn University, and they made some security changes, according to my Global Water Watch contact, which caused these program crashes. And so they've been working on them, but for about, I don't know, a month or two right now, when you try to log data, you get this error. And so it's hard to say, well, did my data actually get in? I guess you can view it later and see if it's in there or not, but obviously there's something that needs to get fixed. So my apologies if this has been frustrating and it's taken a while. Last week, what I was told was they were expecting it should be potentially by the end of last week. So I think we're getting close to this being resolved, but we've kind of put our, currently that's what we're using to store our data. So we're sort of committed to the Global Water Watch database for the time being, although there are some alternatives that I've been exploring. Okay, let's talk about bacteria counting. So this is a photograph of some samples that I incubated recently in a nearby series of streams in the Wandering Creek community. And so you can see that there's, a variety of sizes and colors of colonies growing in there. So I just wanted to take a minute to review some guidelines about counting and give you some help, maybe make available to you a guide. So this is one of those dishes in a more close-up photograph. And, when you go through training, everything seems really simple. There's the blue ones, those are E. coli, and there's the pink ones, and they're not E. coli. And then when you incubate a sample, you get other ones, like you get this kind of a gray blob over here, or you get these like teal colored ones. So you get some things that don't look exactly like the blue or the pink. And so micrology does have a color guide. And so this is something that's useful to refer to. Even with this, it can still be challenging sometimes to draw the line between, do you count it or do you not count it? Is it purple or is it pink? But I would say just do your best, try to be consistent using that. And I'll give you an example of counting that I did. So one thing I also do is, this is a lid, just a clear lid I've taken and drawn a grid. So that's helpful for counting if you have large amounts of other coliform. So going back to this example here, this was quote unquote no name creek. And I'll make it a little bit bigger there. So I counted the dark blue ones like this as E. coli. Some of these look more purple in the photograph than they did in real life. In real life, they looked more pink. And I did not count the teal ones as E. coli. So the count that I came up with is, whoops, sorry, I'm trying to make this bigger. So I came up with, in this sample, I came up with 14 that I counted as E. coli.
Why didn't you count the teal ones?
Because the teal ones are not considered to be E. coli.
So wait, is it the ones on the left are E. coli and the ones on the right are not?
Exactly. Yep. The column on the left is E. coli, and the column on the right is not. And I can email out an image of this. Like, once again, I've sent it out in the past, but I'll send this out again just to help give people something to refer to.
So Eric, you counted the purples as E. coli, is that right?
You do count these dark purple ones as E. coli, but if they're mostly pink, you don't. And I will say that sometimes if you take a picture of it and look at it later, it doesn't look quite the same as it did to your naked eye in light, because I feel like digital cameras and phones sometimes affect the color. So I think I personally like to count it like actually looking at it. But I know in Thornton Creek Alliance program, they actually prefer to take a picture of it and blow it up on a computer so they can look at it bigger. So you can do that. I would just say maybe verify your what you see in person with, compare that to your digital image to make sure they look the same.
Wow, like the dark blue with teal halo and the teal with teal halo, or the teal green, they're really close.
Yeah, yeah.
Yikes. I think I've been, I mean, I haven't been doing the bacteria lately. Jeremy's been doing it, but I think I counted some of these teals that I wasn't supposed to.
Well, I just say do your best. It's a little bit, it's just by its nature a little bit subjective, but I'd say if the same person is applying the same criteria over time, you're going to see trends up and down, and you may be, it's possible you may be off a colony or two.
Yeah, that's true. It's not that much, right?
This chart is really helpful.
It is. I think this is the first time I'm seeing this.
Yeah, yeah.
This is good. Yeah. And sometimes, like the pinpoints, what's too small? I heard sometimes they're so small sometimes that you don't count them at all. Is that true?
So like on this one, like I wouldn't count this one. I wouldn't count these little tiny ones, but I would count like one that's big enough to be, sort of growing as a colony. So I don't count these little tiny dots, but I'll count these bigger ones. Again, that's kind of a, where do you draw the line at a pinpoint? But
Yeah.
officially the curriculum says the period at the end of a sentence. So, and this is obviously this is a little bit blown up here compared to real life.
Okay. I'm glad we're going through this. This is good.
Good. Any more questions on this one? I'm sorry, it's not black and white.
And so are we.
Be harder if it's black and white.
Yeah.
Oh, right.
Eric, on the previous slide, would you just highlight a few of the teal ones that you would have not counted again? Sorry.
Yeah, like this one.
Okay.
So like these are teal, distinctly teal.
Right.
I wouldn't count those, but these are dark blue. So I would count those.
Were there any of the pinkish ones that you did count?
I didn't count these pink ones.
Could you go back to the chart just for a second? Okay. Oh, okay, the pink, purple. Okay, got it.
And Eric, could you show us some purple ones that you did count?
I don't think I counted. I think I considered these to be pink, not purple.
Oh, okay, now I'm really confused because they look purple.
Well, yeah, and that's the thing. I'm sorry. When I looked at, that's why the one downside to this slide, when I looked at them in person, they looked lighter than when I took a picture of them, they looked darker. So it's, I tried to make this as realistic as possible, but they do look more purple in this photograph than they did in person.
Okay.
So I pretty much just counted the darker ones. Trying to see if this would be. Yeah, you can't see it very well there. Okay, any more questions on bacteria or comments? Okay, let's talk about some upcoming training. So we're going to be doing a whole new series of classes for new monitors, but you're welcome to join into any of these as refreshers. So this is the schedule coming up. And I'll put this on our website as well. So if you wanted to attend one of these upcoming practicals, or if you wanted to help teach one of these upcoming classes, or if it's a class that you've never had, you're welcome to take it. So I don't know if everybody has had stream biomonitoring, for instance. Probably most people haven't had BIBI. And I think I will talk about what each of these are. So water chemistry is use of the Lamont kit. So everybody's pretty familiar with that one. This is the physical chemical class, air and water, pH, et cetera, et cetera. So this is something that we're going to do as a Zoom class and then an in-person class. And I'm actually scheduling 2 in-person sessions for this one. I should go back to the whole schedule at the end here. Stream biomonitoring. This is where you learn how to collect and sort benthic macroinvertebrates. And then you identify and categorize them by pollution tolerance. And we'll develop a water quality index based on the proportion and quantity that you have of pollution tolerant or pollution intolerant. And I'm doing this class in the springtime this year because this is something that you can do in the summertime. Really, the best time to do this is August through early October. And again, this is a qualitative method. So it gives you a sense, like if you do it repeatedly at a given site, you can say if it's getting better or worse, but it's not a quantitative method where you could say, well, I got an index of this at this particular site, and I got this different index at the site, at a different site nearby. It's not completely, I should say, it doesn't give you a numeric comparison between sites like another technique that's used by agencies called BIBI. So BIBI, you use a very specific set of techniques to, again, collect benthic macroinvertebrates. So you primarily use a specific type of a net with a way to collect them. And then you preserve them in alcohol. You send them to a taxonomic lab. They come up with a particular score, and that's entered into a database called Puget Sound Benthos. And this is a quantitative method that's used by agencies. And so we're trying to, for creeks that we monitor where this isn't already done by some agency, we're looking to do this on some additional creeks this year to help contribute to the quantitative knowledge about what's going on with the benthic macroinvertebrates in our streams. And feel free as I go through, if you guys have questions about any of these classes, to ask me about them. Another thing that we're going to add this year, this is something that I've been thinking would be interesting and somewhat valuable, is stream flow assessment. And it's pretty straightforward. The basic technique is you calculate the cross-sectional area of a stream at 2 points, an upstream and a downstream point. And then you calculate, you measure the distance between those points, and you time how long it takes a floating object to go from one point to the next. And then you use all that information to come up with a calculation of cubic feet per second. And so that is something that could be useful to either just compare the size of a stream or the magnitude of a particular stream, or to see what's happening with the flow in a particular stream over time. So this is a technique that I've been thinking would be interesting to add. So this spring, we're actually going to add this technique and have a class on it. And then the other thing is There's a program, Pierce Conservation District has a program called Stream Team, and they're a very similar program to ours, but they're just in Pierce County. And they have some great habitat assessment tools. And so in terms of habitat assessment, what we're looking at for habitat assessment is things like characterizing the riparian vegetation around the stream, characterizing the shade, looking at large woody debris in the channel, looking at the nature of the stream bed itself. Is it sand? Is it pebble? Is it cobble? Or is it boulder? What's the nature of the bottom? Or is it muddy? Or is it rocky, etc.? So there's a number of different things that you can use to kind of capture what's going on with that particular reach of the stream. So that's another thing that we're going to be adding a class on. And I think we're going to do this in conjunction with the stream flow monitoring class. So let me just go back to the schedule. So I'll send out an email with this schedule as well. But if any of you guys are taking notes, or you can look at the recording later, those are the trainings that we're planning on offering this spring.
Have you chosen a place for the practicals?
So I think at least one of the practicals, a great location for that is Wallace Swamp Creek Park in Kenmore, just because it has a really nice reach of stream that is, it's also a great place for a bunch of people in class to stand around next to the stream and access the stream. But I think another location that we may do, like the BIBI, is looking at doing Denny Park in Kirkland. It's on Lake Washington, and there's a stream that runs out. And that's one where I don't think BIBI assessment is done. And it's also a great location for people to be able to get close to the stream. But I'll send out locations as well.
You know, Eric, you might also want to teach a technique for doing stream flow from a flowing pipe,
Oh,
because
sure.
that's something that people do come in contact with, and it's a simple protocol for determining flow from if you're able to get the right kind of pipe where you can collect water from it as it flows out.
Okay, maybe you can follow up with me about that after this.
I will not, I could before, just, I'm not myself going to be able to attend any of these trainings because I'm going to be out of the country for most of this period.
Sure.
But I'm going to let LUC folks know.
Okay.
Hey, Eric, are you going to record any of the Zoom meetings so we could access them at a different time?
Yeah, actually, that's a great question. All the Zoom classes will be recorded, and then I'll post links to all these trainings so people can go to the website, get a link to any of these training classes, and they'll just be on a YouTube channel, YouTube link. But then actually one other interesting thing is been working with a partner organization, Cascade Water Alliance, and I think they're actually going to partner with us and a video team, and they're going to record some of our practical sessions as well, so that people can see the practical stuff being done.
Is it for me, Eric, can I just have anyone who's interested in a class email you? Do you have a registration you want to use, or?
Yep, just have them email me.
Okay.
Sno-King Watershed Council at gmail.com, and then I'll create a list of people, and I'll communicate directly with all the people who are taking classes.
Okay.
Eric, I'm interested in that DNA sampling project. Do you anticipate doing a training on that at some point?
That's a great question. I hadn't really been thinking about doing that, but yeah, that's a good question. The project is gonna take place over probably several months to a year. So probably initially what we're gonna be doing is working out some of the bugs in the process and just getting it down to a systematic process, because we're partnering with King County right now. Gary Olson and I are going to go out and capture water samples at locations that we think will be interesting locations, ideally something where there's some bacteria to find. And then we're going to go to King County and partner with them. Initially, they're going to help us filter and extract the DNA, and then that gets sent down to this company Veriset in California that does the sequencing. But there's a little bit of just getting our exact procedures worked out before we're ready to before we'd be ready to share that. But that's a great question. I could see about making that into a class. It'd be an interesting class.
Thanks.
Sure. Okay, any more questions about upcoming training? Okay, then I'll skip ahead.
Oh, we'll just do things. Renton also is a good place for both the stream flow, because the park behind the community center has lots of space for people and we can have access to both sides. And there's also the access where the fisheries department does tagging and stuff. So that's just another possibility.
Okay.
And then with the biomonitoring, I'm excited about springtime, because in the past, when we've done it later, the salmon aren't feeding, but now if you do it in the spring, it'll be perfect, because then when you have the chum coming down, when you have the juveniles going out to sea, they'll still be eating, and we could see if the critters that we're catching are things that they'd actually be eating. So I'm looking forward to that one.
What's the creek in Renton, Debbie, that you're talking about?
Cedar River, it's not really a creek, it's a river. It's big,
Okay.
maybe too big, but it's the major salmon. I mean, it's, you know, they're narrower spots, but it may be too big for it. But in any event, you can come down and visit sometime and I'll show you where I've
Sure.
been testing, where some possibilities are. Unfortunately, the homeless have moved in along most of it, but that's another issue.
Sure. Okay.
Thanks.
Okay. Another thing I wanted to touch on is monitoring timing. So as far as what's a good time to do your different types of assessment. So chemistry and bacteria, we encourage people to do that monthly. In some cases, there's people do sites quarterly. It's really whatever works for you and whatever makes the most sense for you and whatever you want to use the data for. So we recommend monthly. Biomonitoring and BI/BI, we typically only do once per location. And the best time to do that is kind of late summer and fall. So obviously the other thing we need to watch out for is we don't want to disturb salmon reds. So we have to be sensitive that if we're doing this in a salmon stream, that we don't do it during the salmon window. But aside from that, the greatest abundance of these benthic macroinvertebrates is typically in late summer and fall. Flow is typically done anywhere from late spring to fall. You can do it during the wintertime as well. The caveat is that streams can become extremely flashy in the late fall and in the wintertime. And like our partner, one of our programs that we've learned from the Pierce Conservation District Stream Team, they basically don't do any flow monitoring from late October through, I think, somewhere in the spring, because of just a safety issue. They don't want people standing in rapidly flowing creeks trying to measure flow. So that's basically what determines it. But if you could do it safely, really any time of the year would be interesting to determine flow.
Hey, Eric.
Yeah.
So are you taking questions as we go, or do you want me to wait?
No, go, feel free.
When you say flow, are you measuring, what are you measuring? Is that just cubic feet per second, or are you actually measuring volume of flow?
It's cubic. Yeah, I mean, you're trying to come up with a calculation for cubic feet per second.
Cubic, okay.
Hey, Brian, there was some information earlier in the session about that you want to see.
Oh, okay.
And Eric said that you're recording this session, Eric, right? So that somebody
Perfect.
tuned in late could catch up on that. And I've also got notes which I'll be sharing with you.
Thanks, Mark. Habitat assessment, also, again, that's typically done once per year. You could do it more often though, if you want to characterize your stream reach at different times of year. But generally speaking, that's something you compare year to year. All right, so that's kind of recommendations on timing. And does anyone have any questions on timing? Okay, so supplies. Remember when you're monitoring to check for expired reagents or broken glassware before you monitor. And if you get your supplies from us, get in touch with me, unless your program buys its own. Currently, Eric Strom is our supplies coordinator. So if you already have Eric's contact, you can get in touch with him, but typically he keeps me informed on where we are on supplies, and we always maintain a little reserve of supplies. So if you need supplies, get a hold of me or get a hold of Eric Strong. And he notified me that we've got some things that are going to be expiring soon. So we may be getting in touch about replacing some things as well. And as always, thank you guys for the work that you do. Thanks to all the people who support us. Fun monitoring. So I'm going to stop the share at this point and see if anyone else has any other questions.
Hey, Eric, this is Jeremy. Quick, small question. We ran out of data sheets, and I'm not sure how we've handled that in the past. Do we just print them off, grab some right in the rain and print off a bunch, or is there a official way to download them out of the system?
I'm going to be getting more. I just got a big pack of right in the rain paper, and so I will be printing more data sheets. And so if you like the pre-printed data sheets, I can get you those. I can get some printed out for you specifically. Otherwise, you can just use a blank data sheet, and really you just need to write in your site location and your site number and the other information on that data sheet, and then you can just use a blank data sheet. But in either case, just let me know if you want the pre-printed or just a blank data sheet, and I'll get you more data sheets when you need them.
Awesome, thanks. Yeah, blanks are great. Thanks.
Hey, Eric, any thoughts on, I mean, this is me, a non-technology guy speaking, but any thoughts of putting these things on an app? It seems like we're getting into that technological age here. We've had many days of rain where we can't even see what we're writing. And I guess it'd be fun to maybe have an app that you could just kind of plug your numbers into and it saves somewhere, or maybe even uploads directly to the website.
Well, Eric, WaterReporter will do that for you.
Yeah,
I'm ready actually almost to show you what we've done with WaterReporter. It'll be ready by the end of the month.
Oh, good.
Really?
Good. I'm glad you've been using it. So Anna and I were exploring using WaterReporter. That's another data recording application that she was familiar with. And so we started working with the group at WaterReporter. And the plus with WaterReporter is that you can enter the data on an app and that you control the data. And there's different ways you can display the data. The minus on it is that if you were to use, we would have to essentially, if we just went to WaterReporter, we would essentially have to either do some sort of conversion process from Global Water Watch to WaterReporter, or if we wanted to do both and use both, we would have to do some sort of double entry process as people collecting data, which wouldn't be completely ideal. So it's almost like for a given site, it's better to go one or the other. And the one thing with the Global Water Watch that they do is they build in some of the calculations, like if you enter a number of, like bacteria data, it averages that, or so there is some calculations that you would have to do the calculations yourself before you entered the data into WaterReporter.
You mean like for bacteria?
Yeah.
Yeah, that's true. But it will, I mean, that's the only calculation you do, right? Bacteria.
DO saturation, I think also.
Some very minor ones, right? The number of drops gets translated into,
Oh, yeah.
into a number.
Yeah.
In 2 cases, but that's pretty
Yeah.
minor.
Yeah.
Yeah. So great.
You do have the ability to batch load. Like you could collect data for 2 or 3 months. And then as long as you have a standardized data entry form, you can batch load it all up into WaterReporter. And I mean, it's Serge, right? Serge. He was able to batch load some of my data. So you could, instead of doing data point by data point, survey day by survey point, you could make an agreement to batch upload all your data for a period of time to either WaterReporter or to Global Water Watch.
Anybody want to be my data coordinator?
Well, it would still be, everybody's responsible for their own data, right?
Yeah.
I think the only thing you would need to do is create a template for what they would have to enter their data into a spreadsheet. And then they they would batch upload their data they could just email it to Serge, or they could email it to you and you can batch upload it. It's really pretty fast.
Into WaterReporter?
Yeah, it's both. I mean, they might, like, Serge might say, well, I need the dissolved oxygen to be listed before the pH. But with WaterReporter, you decide. That's the part of the WaterReporter I have not yet figured out. That's allows you, and it allows you to kind of create any kind of survey you want to create. So for example, I know that some WaterReporter groups in the Chesapeake Bay simply use a simple WaterReporter form for people to enter information about like seagrasses, like taking certain measurements and whatnot. They enter it out. I don't know if anybody the only thing I don't know yet is how complex a form WaterReporter can handle. You're still, it is nice to have a data sheet as a backup. It just really is nice. And I like to have a data sheet as a backup. It's easier to work on than a little tiny mobile phone screen. And then when I come in from the field, I just enter my data into a spreadsheet. Well, actually, I have one of the youth in my program enter the data into a spreadsheet and I check her work. And then I've got, 2 years of data I could batch upload in one 5-minute session on the WaterReporter. And then it's pretty simple.
Do you use Excel, or what do you use to enter your data into?
Well, I keep my data because I have another youth who's accessing the same file. I keep it as a Google Sheet.
Sure.
And then when I want to batch upload it, you essentially export the Google Sheet as a comma delimited file, a CSV file. Mm-hmm. Usually that's what they want when they're uploading into an online database.
So can you make a template available for us?
Well, a template with WaterReporter, you decide what you want to enter in to WaterReporter. So there's certain things on the WaterReporter, on the Global Water Watch sheet. There's a lot of data there that you may not want to put up on WaterReporter. You might only want your site code and the actual data, but like there's stuff about weather conditions, there's observational data that you may or may not want to put up in WaterReporter. So it's really up to you to decide what you want to show in WaterReporter. You might say, you only want to show oxygen temperature, or you want to put all your parameters up. But sometimes it's only valuable because WaterReporter will be able to graph the data over time. as you put more and more data for that one site, it'll be able to graph the data. It'll even be able to do in theory, they have the technology to do a report card for your stream to say what's failing and what's passing. But it's up to you to decide what you want to track. And for example, there's no state standards for alkalinity or hardness. Do you really want to upload that data to WaterReporter?
But if you don't have the data set up right when you batch upload,
You do
then
have to,
things have to appear in the right columns, or it's
Yeah.
not going to know what to do with your data.
Yeah.
So is there like a template that we could use where we can leave certain columns blank?
yeah, I would think it would be, since you would be, if you're going to use the WaterReporter version that Eric still has, and it's up to you, Eric, whether you're going to maintain that. It would be really up to you, Eric, to decide what data you want to put up on WaterReporter. And you would provide a blank template to everybody. They enter their data, and maybe once a year or once every 6 months, they'd send you their data, and you would just batch upload it all. We might have to do a review to make sure they didn't make any obvious mistakes. Batch upload it, and then you've got all the data points in there, and all the data and all the graphs, and you'd have quite a lot of stuff up there that you could see.
Yeah, I think that could work. I think the one thing that we would also have to do would be develop some procedures around it to make sure that we sort of had a QA process built into the collection and the, just bring it to an Excel sheet. And then, because once upon a time in the past, before we entered our data into Global Water Watch database, what we did is we had a volunteer. People would email. This was still cumbersome, but people would email a scanned version of their data sheet to this central point, this volunteer, who then entered it all into Google Sheets, and we had that information available on our website, which worked. It actually displayed great, and it worked in some ways, but it was kind of cumbersome. But we did have some QA steps built into it. So I feel like if we do move to the WaterReporter, we'll have to build some procedures around it and some QA steps along with it, just to make sure that it's all transferred accurately. Because I think that's part of the Global Water Watch. Theoretically, we're working underneath Global Water Watch's QAPP, Quality Assurance Project Plan, and part of their QAP is that use these data forms, use these procedures, and they have that little online review. You review your data before you hit submit. So I think that actually has a lot of promise. But I think what we need to do is build some procedures, and I would actually need to find some volunteers who would be willing to work on this project, because I don't personally have the capacity to really do that.
If you're willing to let me take your screen, I can show them what I've put on WaterReporter real quick.
Sure. Let me you should be able to share it now.
Okay, so like that. Make that big. So this is we're only doing Springbrook Creek watershed. So I have a few inactive stations that are showing as gray. I haven't, this is not for public consumption yet. Some of my sites aren't lining up properly, but that's my own issue. But if I go in and I look at a site, I have 2 points here. I can see all of my data for the last 2.5 years, really. And I can go through and pick an individual parameter. So let's say I want to look at oxygen, and I look at this site, and it's telling me I have a photograph of the site. I can get the last measurements that were done, and I can see the trends for different things, and I can look at oxygen. I'm still, like, as I said, working this out, but it's giving me the basic thing, and I haven't I can color code them to turn red if they fail, or green if they're, or blue if they're passing, or yellow if they're on the cusp or something. I've got those kind of controls, but this is just a basic, this is basically me batch uploading the data in a raw form and not having really done much else with this table since I did that batch upload. But I can look at temperature and see, you know, data for temperature. I've got Mill Creek 2.0 is failing. And I can just go through the data and take a look at what's failing. And so that gives you an idea. Here's my trends.
And when you say it's failing, are you setting the target parameters?
Yeah, you can see the target is if the temperature is higher than 17.5 degrees Celsius, which is the state standard, then it's failing for water temperature. So this reading, the last measurement was 18.3.
Okay. So you did you input those, or were those?
This was
Okay.
batch uploaded data, and it's 2 years worth of data. So,
But like the pass fail, like the
yeah,
target.
it's a pass fail. Yeah,
And you put those in?
yeah. I tell
Okay.
the system what's passing, what's failing.
Okay. Yep.
Only so for oxygen and temperature, those are the only two. pH has a pass fail, but turbidity doesn't. I can make one. I could say, well, anything with turbidity, like I know that this is a very turbid site, and I don't know if I set a pass fail for turbidity. Temperature. Oh, I did set a pass fail for it. So yeah, because the state standard is like 5 NTUs, and I'm using a turbidity tube, 5 NTUs over baseline. So for me, I considered historical data on my creek where 10 was the historical baseline. So anything over 15 would be considered failing. But a lot of these specific conductants has no active thresholds. Nitrates, you can categorize them as just fair to excellent. There's no state standard, but I had a, you know, somewhat of a scale for that. I think it's over 3 and it's considered starting to be poor. And so that's kind of an idea of how it works.
Hey, Anna, do you
I
have
don't have status yet because I haven't set a report card for this. Yeah.
Anna, do you have one of the Google sheet that you can show us too, the kind of Excel file that you would normally
Yeah.
enter your data?
Let me stop sharing for just a second. Switch over to that. Drive is right. See, monitoring data. My backup data sheet. So this is my data sheet. And it follows pretty much the Global Water Watch like list of everything you put in. You know, I do a few extra things, like I take stream depth and stream width, and I put that, those are 2 that I put in. So this is all entered by one of my youth, and you can see my data runs from September 2019. Sorry, still loading. Oh, I'm sorry. I loaded only the data up to 2020. I have another sheet I'm using that goes past this. I've got data up until this past month, but this is basically what it is. Simple spreadsheet using Google Sheets.
And do you load this data while you're at the site when you're
No,
oh,
I take my spreadsheets home, and I actually send them to the youth, and they enter them. And then I retain the original sheets, and then I just review their data entry to make sure they didn't make any typo mistakes.
You take your data sheets home, you mean? That's what you
Yeah,
said.
I take the data sheets back with me. I scan them actually to send to the youth that enters the data. And she reads them and enters the data, and then I go through and do a check, and that's my quality assurance check. And that's just all this is.
Did you make this spreadsheet though, or was this provided to you?
I made it.
Okay.
Yeah, and I just looked at the data sheet and I said, well, the first question is group, and the second question is who are the monitors, and that's what's the address, and it's just all the questions in the data sheet listed one after another. The only thing that's different in mine is that I do width and depth. Width and depth, and I also do I've added we do collect garbage, so I put that in there, and I have a few gauging stations that are on my site. So for the day we're out there, I go and check the gauging station to see what the flow rate was in cubic feet per second. And I enter that information. This is the additional tests if I have them, but all these other, aside from garbage collected, gauging stations, width and depth, all of this is the data that's in the spreadsheet, in the data sheets. These are the columns, the titles, everything that are in the data sheets.
So is this saving you time over doing it individually, entering your data individually as you complete?
I think so.
Okay. Plus, well,
I
you
think
have
so.
somebody to help you too.
But I mean, the thing is, I have only uploaded, I think I only uploaded a year's worth of data. And I did it by I basically gave this data sheet, a year worth of data to Serge, and he uploaded it.
Okay.
And he could go in and say, well, I want pH to come after dissolved oxygen. So he can physically move this column over to the right. If he wants to change the order, it doesn't really matter. You can always reorder, move the columns around.
This is actually very similar to what we did prior to us having our sites set up as in Global Water Watch. Is we collected our data sheets, we sent them to a volunteer. Our volunteer entered the data from the data sheets into a spreadsheet just like that. And then as opposed to using WaterReporter, I think the one component that WaterReporter gives us over that is the mapping capability. But
well, but that's what WaterReporter would give you.
right, that's what I'm saying, is that's
Yeah.
the, but otherwise it's very similar to what we used to do. So
The other thing is that it'll have this ability to you can program into WaterReporter the thresholds. So it can tell you when your data's failing, and it can also take a look at all the data you enter over the course of many years and create trend graphs for you, so you can see if things are getting better or worse or staying the same.
let me share one thing briefly. So this is our Water Watchers page, and we actually have a link of this is back when we had our, prior to us converting to Global Water Watch's database. And so we basically had our spreadsheets go to a page where it showed information about the location. Let's see, monitoring.
Oh
And
yeah,
then
nice.
So we basically had graphs.
You
and
could put a picture in. Yeah.
So we're basically doing a very similar thing prior to converting over to the Global Water Watch database.
Yeah.
So it seems like I guess there's multiple ways to do this. We kind of made a decision I think in 2019 to kind of go full in with the Global Water Watch technique. But if we're going, with the programming issues we've had recently, it's worth evaluating if there's some other ways that we want to manage our data.
Yeah. I'm gonna have to apologize. I have to sign off, but I'm still tweaking my WaterReporter, but it's getting closer for me.
Okay, well, thanks, Anna.
All right, take care, everybody.
All right, well, interesting discussion. I mean, part of it is the data collection process where you're out there in the field, and then part of it is the data management. And that's a big part of it. So I don't know if anybody has a special passion, desire, or wish to be our data management lead, but I would welcome anybody who wants to relook at this and maybe take a leadership role in this data management. And I'll send that recruiting message out in our recorded Zoom meeting as well.
Eric, I'll talk to my friend Jill and see if she's interested. Get you two guys to talk.
Sure. Yeah, that'd be great. Yeah, because I think that Anna brings up some great points about there's some good features of that water reporter. But there's also good features of Global Water Watch. It's convenient for the monitor who goes out and does their monitoring, and then they just come back and they sit down and they do their entry, and then it pops up right there and they can see it, and it is integrated with Global Water Watch's system. So there's definitely, but it sounds like there's also options that we could, people could do it one way or the other and have the data transfer over in some sort of a batch process.
I also think you need to think about your audience for the data. I mean, who's looking at this? Where are they going to go to find it quickly? I'm working with a Mill Creek community. They want to be able to see the data, so it needs to be in a place where they can easily find it and understand it.
Mm-hmm. Yeah.
And I know that if anything involves my putting something into an Excel sheet, I have no idea about Excel sheets. I'm good with paper and pencil and maybe uploading, entering stuff. But if it comes to my doing something in an Excel sheet, it ain't happening. I mean, it just is, it's beyond me. And I even took a class once to try to understand. It was like, what the heck is this? So that's where I'm at.
All right. Well, does anybody have any parting thoughts? I'll just say thanks for everything you're doing. And for the time being, we're sticking with Global Water Watch database. We're hoping to get it up and, get it fixed, but I would certainly be open to exploring other options. And if people want to help out in that process, that would be welcome.
Well, hey, Eric, I just a follow-up question. This kind of last discussion started with the comment about how we could keep our paper data sheets from getting drenched in the rain. And so that led to the discussion about an app. But I think you mentioned earlier that you have Write in the Rain paper.
Yes.
You have our sheets that are available printed on that. I did not realize that.
Yeah, I don't know if earlier in the meeting I mentioned that. So
I know you did, yeah, but I hadn't heard it before. I just wanted to be sure. So we can ask you for just a quantity of those. We have 4 sites here.
Yes.
Okay.
Yep. I'll get those printed for you.
Will that paper handle okay, like this for a scanner? Will my printer scan it okay and handle like regular paper?
Yeah.
Okay. Thanks.
All right, you bet. Okay, any final thoughts? Anything else? All right, well, thanks, everybody.
Okay, thank you, Eric.
Thanks, Eric.
Thanks, Eric.
Thanks,
Good
Eric.
night.
And everybody.
Bye.
Bye.
Thanks, Eric.